CRISPR-Cas mediated homology-directed repair, or HDR, can precisely introduce desired sequence modifications to the genome. This can be used to ask biological questions or even cure genetic diseases. High HDR rates are needed to correct mutations, but achieving this can be challenging. Even more puzzling, some cells are easy to edit while others are hard. We use genome-wide screening in Fanconi anemia patient cells to uncover suppressors of CRISPR-Cas HDR. We found the exonuclease TREX1 is a key factor affecting HDR when linearized DNA is used as a repair template. TREX1 expression acts as a biomarker for HDR efficiency, since its expression reduces the levels of HDR. We furthermore identified strategies to enhance HDR outcomes, including TREX1 removal and using chemically protected DNA templates. Our findings provide important implications for advancing HDR-mediated genome editing in research and therapeutics.